Good morning, students. So now we are going to deal with some fundamental concepts of enzymes. So what are enzymes? So you might have come across in your lower classes regarding the enzymes. Enzymes are biocatalysts, the catalysts of life. So what is a catalyst? It is defined as a substance that increases the velocity or rate of a chemical reaction without itself undergoing any change in the overall process. What does this mean? So with an example, a good teacher is always a good catalyst in student's life. So you understand very well about this example. So you, as you grow from your childhood to your college and postgraduate level, so you come across many teachers. So they will be teaching the same thing to all batches. Students will be going from one class to the next level, whereas teacher stays there only without any change. So similarly, enzyme is also a catalyst. Without undergoing any change, it will increase the velocity or rate of a chemical reaction. Okay. So enzymes may be defined as the biocatalysts synthesized by the living cells. They are protein in nature, exception RNA acting as a ribosome, and colloidal and thermolabile in character and very specific in their action. So another example about enzyme is in the laboratory, hydrolysis of proteins by a strong acid at 100 Celsius degrees takes at least a couple of days. The same protein is fully digested by the enzymes in the gastrointestinal tract at body temperature, that is at 37 Celsius degrees, within a couple of hours. Isn't it? So enzymes play a very important role in our health and life, without which we cannot survive, without which our metabolism comes to standstill. What is the historical background? So Berzelius, in the year 1836, he coined the term catalysis. That means in the Greek, catalysis means it's to dissolve. In 1878, Kuhne used the word enzyme in Greek in yeast to indicate the catalysis taking place in the biological system. Isolation of the enzyme system from cell-free extract of yeast was achieved in 1883 by Buchner. So he named the active principle as gymase, later found to contain a mixture of enzymes which could convert sugar to alcohol. So James Sumner first achieved the isolation and crystallization of the enzyme urease from the jack bean and identified it as protein. So here on the right lower corner of the screen you can see the jack beans. So coming to nomenclature, each enzyme is assigned two names. The first is its short, recommended name convenient for everyday use. The second is a more complete systematic name which is used when an enzyme must be identified without ambiguity. So it is something like how do you call neem tree? Neem tree in English, it's neem in a common colloquial term, whereas in the Ajadirachta indica in the scientific name. So similarly, enzymes are also named like that, one common name and one systematic name. So International Union of Biochemistry IUB in 1961, it formed certain rules for enzyme nomenclature and classification. The enzyme has two parts. First part is as the name of the substrate, and second part contains the ending, and A's indicate the type of the reaction. For example, if you take hexokinase, it acts on the hexosis, that's the substrate, Kinase means the type of reaction. Okay, so only exception to this kind of nomenclature is pepsin. So here pep and sin doesn't fit to this kind of nomenclature and classification. So most commonly used enzyme names have suffix ase attached to the substrate of the reaction. For example, glucosidase and urease. That means it acts on the glucose and urea respectively, or to a description of the action performed. For example, lactate dehydrogenase. That means it increases the rate of lactate dehydrogenation reaction. And adenylyl cyclase. So it is a cyclization reaction of the adenine ring. So some enzymes retain their original trivial names which give no hint of the associated enzymatic reaction, because these were very popular before they undergo nomenclature. So those names were kept like that only, like trypsin and pepsin. In a systematic naming system, enzymes are divided into 6 major classes, each with numerous subgroups. So for a given enzyme, the suffix ase is attached, including the names of all the substrates. For example, lactate, nicotinamide adenine dinucleotide oxidoreductase. So each enzyme is assigned to a classification number. So lactate NAD plus is 1.1.1.27. So the systematic names are unambiguous and informative, but are frequently too cumbersome to be of general use, because you can remember lactate dehydrogenase, but you cannot remember every time like lactate, nicotinamide adenine dinucleotide oxidoreductase. It's very difficult to remember such a long name along with this number. So here are some enzyme classes with the example and their reactions catalyzed. Here I give 6 enzyme classes: oxidoreductases, transferases, hydrolases, lyases, isomerases, and ligases. So as you all know, oxidoreductases, the example is alcohol dehydrogenase, cytochrome oxidase, and amino acid oxidase. So what is the main function of these enzymes? Oxidoreductases, they take over this oxidation to reduction and reduction to oxidation. So transferases, example is hexokinase. So you can easily see there is a group transfer is involved in these transferases. So if you take hexokinase, the transfer of the phosphate group, making it as a glucose 6 phosphate, for example. Then coming to hydrolases, example is lipases, or cholinesterase, or acid and alkaline phosphatase, pepsin, urease. So they break down the compound with the addition of the water. These are hydrolases. So lyases, example is aldolase, histidase, fumarase. So what is the function of the lyases? The addition and elimination. Isomerases, triose phosphate isomerase, phosphohexose isomerase. So they just simply interconvert the isomer forms. Then the last one is ligases. Example is glutamine synthase and carboxylase, acetyl-CoA carboxylase, succinate thiokinase. So the main function is condensation. Usually it is ATP dependent reactions. So what are the properties of enzymes? Active site. The enzyme molecules contain a specific pocket or cleft called the active site. The active site formed by the folding of the protein contains amino acid side chains that participates in substrate binding and catalysis. The substrate binds the enzyme non-covalently, forming an enzyme substrate complex. So here you can see the active site and the substrate binding into the enzyme. So binding is to cause the conformational change in the enzyme. So whenever there is a substrate binding at the active site, it will lead to the conformational change in the tertiary and quaternary structure of the enzymes. So this is what is called induced fit model. So which allows the enzyme catalysis. So enzyme substrate complex is converted to an enzyme product EP. So here you can see in the third and fourth part of the diagram, this enzyme product complex that subsequently dissociates into enzyme and product. Thus the product leaves the active site of the enzymes, so that the enzyme which has undergone a conformational change, that will regain its shape back, and it is very much ready to take over another substrate at this active site. So chemical nature of the enzymes. All the enzymes are invariably they are proteins, but all proteins are not enzymes. But all enzymes are invariably proteins. But in recent years, however, a few RNA molecules like ribozymes have been shown to function as enzymes. So here you can see the protein primary structure with amino acid peptide bonds here, which will give rise to secondary structure, and further they will fold into tertiary structures, and ultimately you can see the 3D structure, that is quaternary structure of the enzymes in this ribbon diagram. Okay. Chemical nature of the enzymes. Each enzyme has its own tertiary structure and specific conformation, which is very essential for its catalytic activity. The functional unit of enzyme is known as holoenzyme, which is often made up of apoenzyme, that is the protein part, and the coenzyme, that is the non-protein organic part. Here in this diagram you can clearly see the apoenzyme and the coenzyme part, which is around the catalytic site and along with the cofactor. The conjugated enzyme or holoenzyme is formed by the apoenzyme plus cofactor. So the non-protein part of the conjugated enzyme is called cofactor. The cofactors are of three types: prosthetic group, coenzyme, and activator. So what is this prosthetic group? When the non-protein part attached with the apoenzyme is an organic compound and is firmly bonded with the protein part, the apoenzyme, without denaturation the protein. So example is cytochrome. So here you can see the apoenzyme along with the prosthetic group. So in this cytochrome ribbon diagram, you can as well see the heme part, that which is forming the prosthetic group of these cytochrome enzymes. Second one is coenzyme. When the non-protein part is an organic compound and is only loosely attached with the protein part, it is called coenzyme. So the examples for the coenzymes are NAD, FAD, NADP, etc. This coenzyme, because it is loosely attached, it can be easily separated from the protein part and can be easily attached again with it. So here in this enzyme alcohol dehydrogenase enzyme ribbon diagram, you can see the coenzyme binding part, and the coenzyme is NAD, nicotinamide adenine dinucleotide. So third one is activator. So when the non-protein part is an inorganic substance such as some metal ion or mineral ion, it is called as an activator. The main role of the activator is to form a bond between the enzyme and the substrate molecule. Example is potassium, copper, manganese, iron, zinc, and calcium. For example, if you take carbonic anhydrase enzyme, it has got zinc as the activator. So coming to the types of enzymes, the term monomeric enzyme is used if it is made up of a single polypeptide, for example ribonuclease, trypsin. So these are single polypeptide proteins. Some of the enzymes which possess more than one polypeptide chain are known as oligomeric enzymes. Examples are lactate dehydrogenase, aspartate transcarbamylase. So there are certain multi-enzyme complexes possessing specific sites to catalyze different reactions in a sequence. So whenever you come to the intermediary metabolism, like citric acid cycle and fatty acid metabolism, then you will definitely come across such a big enzymes, that is multi-enzyme complexes, like the multiplexes you see in the shopping malls. So they have different kinds of active sites embedded in their quaternary structure. So only the native intact multi-enzyme complex is functionally active and not the individual units if they are separated. For example, pyruvate dehydrogenase enzyme complex, fatty acid synthase complex, prostaglandin synthase complex, etc. So the enzymes exhibit all the general properties of the proteins. So coming to the advancements in the fundamental concepts of enzymes, the genetic engineering and the modified enzymes or artificial enzymes. So recent advances in biotechnology have made it possible to modify the enzymes with desirable characters, improved catalytic abilities, activities under unusual conditions. This approach is required since the enzymes possess enormous potential for their use in medicine and in industries. So hybrid enzymes, it is possible to rearrange genes and produce fusion proteins. Example, a hybrid enzyme of glucanase and cellulase that can more efficiently hydrolyze barely beta-glucans in beer production. So site-directed mutagenesis. This is a technique used to produce a specific mutation at a predetermined position in a DNA molecule. So the result is incorporation of a desired amino acid of one choice in place of the specific amino acid in the enzyme. That is, example, tissue plasminogen activator used to lyse blood clots in myocardial infarction. In recent years, it has also become possible to produce hybrid enzymes by the rearrangement of the genes. So another concept is AbGenzymes, that is antibody enzymes. So these are also called as catalytic antibodies. Okay, this finishes the fundamental concepts of the enzymes. Thank you very much.